Welcome to SNN.Network Canada virtual event. I'd like to introduce our next presenter, Mark Williams of Marvel Biosciences, listed under TSX Venture under the symbol MRVL. Sir, the floor is yours.
Thanks very much, and thank you to everyone participating this afternoon. This is our disclaimer, which you can read at your leisure. I apologize for the slight delay. So briefly speaking, Marvel is a biotechnology company focused on developing essentially known derivatives of known drugs. We call this process drug redevelopment. And because of that, we believe as a company that we can significantly lower the cost, the time, and the risk associated with bringing a new chemical entity to the market, because we know the original toxicity, we know the target, and we know elements of the drug that allow us to do that very, very quickly. Between us, we have, as management team, more than 50 years of experience. We have 2 compounds that we're working on right now in 3 major disease areas with an aggregate value of roughly $215 billion. This idea of drug redevelopment, that is taking a drug and modifying it slightly, is not unheard of. We're not the pioneers in this space. Certainly an example that some people might be familiar with is the very large biotech, US biotech Celgene. They took a drug called thalidomide, which was originally used to treat morning sickness. It was obviously vilified and withdrawn from the market, but it was found to have also anti-cancer activity. So Celgene re-engineered the molecule and chemically modified it so that they had less toxicity associated with the drug. And that is currently a billion-dollar blockbuster drug for that particular company in the name of Revlimid. It also allowed them to get new composition of matter patents and a 20-year life time on that molecule. A Canadian example of a company that's successfully done this is Aurinia. They took a drug called cyclosporin, originally used for the treatment of transplantation, made a very, very conservative small chemical change to it, improved, I believe, the kidney function or reduced kidney toxicity. That became the molecule voclosporin. I think they got approval in January for the treatment of lupus nephritis and were recently the subject of a takeover bid. So our company and its lead asset is MB204, which is a redeveloped derivative of istradefylline. And we're looking specifically at the use of the molecule for depression and Alzheimer's diseases, so neurological diseases, but also a non-neurological disease, non-alcoholic steatohepatitis, NASH, which is fatty liver disease. And that is a non-CNS disease. So we're trying to have multiple uses of our asset in order to de-risk its development. So if it fails for one indication, it may have applications and use for another indication yet. MB204 is based off istradefylline, as I mentioned, and istradefylline was originally approved for the treatment of Parkinson's disease. It's highly related to caffeine, but it's about 100 to 150 times more active than caffeine, and it works by modulating the dopamine D2 receptor. Interestingly, the target of the adenosine A2A receptor, and in fact istradefylline as well, has been associated with multiple other diseases. So this is the idea of taking an old drug and using it for a new indication, and in this case we're slightly modifying it. So there's very good evidence from the scientific literature that this target, the A2A receptor, which is what our drug goes after as well, is involved in depression and anxiety. There's evidence that it reduces fibrosis, particularly in the liver. And there's good interest now in the same target for neurodegenerative diseases, specifically Alzheimer's disease. And that date is circled here because it's basically the last week of November where this recent paper came out. So I think it's, given the success of Biogen and its Alzheimer's drug and the reinterest now in the Alzheimer's space, we think we're poised to take advantage of that and are going after a new target for the treatment of Alzheimer's with this molecule. So Marvel has screened multiple different versions of istradefylline and has settled upon its lead asset, MB204, which is a fluorinated derivative of istradefylline. We filed for composition of matter patents in March of last year. Our lead molecule is very similar in its ability to go after the target as measured in cell cultures using what's called in vitro IC50, so the numbers are very similar. on the left-hand side, but on the right-hand side, we are showing that the molecule, with the changes that were made, these extra fluorine groups, the molecule appears to be better absorbed, that is, it gets to higher blood levels, and tends to last longer in the bloodstream. So it looks like it's better bioavailability and a longer half-life is how that's explained. So our first indications are CNS related, and this represents at least a minimum of a $30 billion market. One can argue that it's substantially larger than that, especially with the renewed interest in Alzheimer's disease. One of the concerns of Alzheimer's is that 1 in 3 people roughly over the age of 70 has got some form of dementia. So with the aging population that we have and the life expectancy increasing, especially as we become better at treating heart disease and cancer, the concern is that the mind will go, and we're going to need armies of caregivers to look after people. So this is often referred to as the silent epidemic. In the case of COVID, another quiet epidemic seems to be the increase in depression and anxiety symptoms in the overall general population. So much so that almost 1 in 2 or 1 in 3 people is complaining of symptoms of depression or anxiety, which is remarkable. This has now gone up about 50% overall as a market, and it's estimated to be at least 30 billion now, with a 50% increase over the last 2 years in the requirements for drugs to treat it. And one of the problems that we have with the current class of antidepressants is that, certainly in the case of the most widely prescribed serotonin reuptake inhibitors such as Prozac, is that they take a bit of time to work, as much as a month in some cases, and about 20% of people don't respond to them. And of the people who do respond, up to 40% of those people tend to report symptoms of apathy and loss of motivation. Loss of libido is another symptom that's often ascribed to people on that particular drug. So much so that people have literally said that they've lost their ambition. And one of the questions is, wouldn't it be interesting if you could take a drug that could actually motivate you as opposed to just keep you somewhat level? And that might actually be the case with this particular class of drugs. Again, going back to the original diagram showing you the mechanism, the istradefylline is able to inhibit the adenosine A2A receptor, which negatively crosstalks with the dopamine D2 receptor. And the reason this is important is the dopamine pathway has been known to be involved with motivation and the reward pathways. And as such, it could reduce the anxiety and apathy associated with loss of motivation. So this could be a very interesting first-in-class treatment for antidepressants. What is known is that istradefylline has been used in Parkinson's patients, and some patients have reported an improvement in their mood and depressive symptoms and the anxiety symptoms in that case. So suggesting that there is some evidence of a clinical benefit already, although the original company probably will not be pursuing istradefylline for these indications because they've run out of patent life, and it's not going to be in their interest economically because there's now going to be generic version shortly. Just to give you a sense, we've as of today started dosing animals for our toxicology and IND-enabling toxicology experiments, but we do have some preliminary evidence that the drug appears to be safe. We have tested it head-to-head against istradefylline and see that we don't see any new or overt signals or problems of the liver as measured by what's called ALT and AST enzymes. You go for a liver function test, they'll often check this to make sure your liver is healthy out of your blood. And there seems to be no negative effect on the kidney at this dosage as shown by the effect on creatinine in the blood. However, the drug does appear to be quite effective, and this is shown in the right-hand panel with the forced swim test. So two things that should be noted is that these animals were given our drug MB204 1 hour to the test, and the symptoms, the reduction in depressive symptoms is dramatic. It's fast. It's within the hour, and there seems to be a very nice dose response with respect to the drug at the highest dose of 10 milligrams per kilogram. And it's important for those people who may not be familiar, those stars that are above the bars there are reflective of statistical significance, and the more stars you have, the more confidence you have in the strength of the data. So the fact that at our 10 milligram per kilogram dose, which is comparatively low actually, we have a very nice, strong, statistically significant signal. Anything less than 0.05, p less than 0.05 is considered relevant and important. So the fact that we're well below those levels is a good sign with this drug. So again, the world is certainly clamoring for new drugs to treat CNS-related diseases, and those have high risk, high return potential. As a company, we're also very interested in de-risking ourselves. So we are intending to find other non-competitive diseases that we can use with our lead molecule. And in this case, we've got some preliminary evidence now in fatty liver disease that our drug may have some potential. And fatty liver disease is estimated at a $20 billion market, although I've read reports that it could be as high as $80 billion, depending on who you read. And really, what the case here is that it's what physicians really get worried about is the development of fibrosis. So this is almost like the scarring of the liver, just like a scar on your hand. When you cut your hand, you can get scars in your liver as well. And the reality is the right-hand panel is showing you that you can live fairly comfortably. There doesn't appear to be any change in your mortality signal really if you've got a fatty liver. But if you have a fatty liver with fibrosis, then your mortality risk goes up dramatically, especially over time. So what it's telling you is that a fatty liver is probably okay to live with, but if it develops fibrosis, this is a major problem. And if you look at the left-hand panel here, where it talks about the epidemiological studies in the U.S., about a fifth of the population has the steatosis, the NAFL, and of that, a subset will go on to get fibrosis, maybe as much as 20% of those patients as well. So, we have a fairly high number of patients at risk of developing fibrosis from the general population, maybe as high as 4%. And if they do develop fibrosis, you run the risk of getting cirrhotic, you get cirrhosis, and possibly liver cancer. All would require likely a transplantation, which would be very, very costly. So this represents another sort of silent epidemic that we're dealing with. That's a consequence of lifestyle potentially and other factors, but certainly associated with aging as well. Now, there are currently no approved treatments for NASH, although there are many in development, but this is a fairly busy slide that talks about all the compounds in development, and it's a bullseye diagram. So the closer you get to the center, the later the stage of the molecules. In this case, I wanted to point out one segment of the group of molecules in development, and that is the anti-fibrotic group. And of the anti-fibrotic group, you can see that there's not that many molecules in development, maybe 8 to 10 at the most, and some of these have actually even dropped off since this figure was generated. The lead of which, and you can see the lead if you look closely towards the center of the bullseye in that one segment, is a compound called cinacriviroc. Cinacriviroc is in phase 3 testing right now, and it's probably the leading anti-fibrotic asset in clinical development. That will be important in the next slide after this. And what we know from the literature is that there seems to be an important signal generated by the adenosine A2A receptor that results in the hepatic stellate cell releasing collagen. And collagen is the component of scars and the fibrosis that we talk about. So when you cut yourself, it's the collagen that forms that scar over that. And a similar process occurs in the liver once it's damaged. It turns out that blocking the adenosine A2A receptor could be a good prevention of fibrosis, and our MBA204, which goes after that receptor and antagonize it, is expected to block that signal as well. So in this case, if you are to pursue a drug for the treatment of NASH, the FDA has two key endpoints. The first is what's called the NAS score, and we studied that in study 1 on the left-hand side here. And the NAS score is a composite of 3 basic endpoints, and then you add the scores up together, and that gives you a final number. So reducing that score is a good thing. And we show here that in the case of NB204, we reduced this NAS score by about 1.4 units, which is okay. It's a modest drop. If you were going exclusively after a drop in the NAS score, then you'd probably want something higher in the 2 to 2.5 point score range. But more importantly, on the second side, on the study 2, on the right-hand side, we're showing you the fibrosis score of our molecule, and this is in a very different disease. And these are 2 different labs, by the way, that conducted these experiments for us. And what we're seeing here is that compared to controls, we're seeing a 47% reduction in fibrosis with our molecule. And interestingly, despite giving a healthy dose of cinacriviroc in these animals, which was known to reduce fibrosis in other models, we see very little effect. And in fact, our molecule is statistically significantly better than cinacriviroc in this head-to-head study. So really, we've kind of, in our opinion, de-risk the development of the asset somewhat, and we are, quickly transitioning from a preclinical stage company into a clinical stage company. So this is some of the milestones associated with 204 that I'd like to point out to you. And as we go forward and start moving these along, we've started to bold these in green to see that we've hit our milestones. So we have completed our preclinical testing in the NASH models, and we're satisfied that there's a strong signal there. We're intending to do a test in an oncology model that we hope to run out, get a data readout within the end of this particular quarter. We are in the middle of the manufacturing of our 204 for clinical trials, and we have just got to the point now where we've got delivery of multiple kilograms of the material that we're going to use for the toxicology testing, and it's what's called the engineering run of the API, or active pharmaceutical ingredient. And so that is coming along very, very nicely, and we expect to complete that shortly. And finally, on the bottom of that panel on the Q4 2021, we finished patenting other derivatives similar to 204 that we think will give us the confidence now to start reaching out to larger pharma partners to introduce ourselves and see if they're interested in our program and our molecules. As we move forward into Q1 of 2022, we intend to complete the cGMP run of the material. That's what's ultimately going to be used for the clinical batches and testing in man, and begin our 4-week GLP, good lab practice, toxicology studies, which you need to complete for man. And by Q3, those studies, toxicology studies, should be complete, and then we will go and approach the FDA and have what's called a pre-IND meeting to go over our data. And Q4, we intend to begin our clinical trial with 204, pending all the toxicology, assuming positive data for that. Our goal is to actually test the molecule and prove it's active in man. So this is kind of an interesting play, because we think we could get an early readout of efficacy in patients by Q1 of 2023, which is normally would take a lot longer, because in the phase 1 you typically do safety, and in phase 2 you do efficacy. So we actually believe that we can show an improvement or a signal in man early on, and we believe that that could substantially increase our valuation. Historically in biotech, the greatest valuation increase often occurs when you show your drug is active in man, and we intend to try and do that fairly early in the life cycle of the drug development process, that is in the phase 1. We would like to potentially use that to seek a U.S. listing, and there's some very good comparable companies at the phase 1 level that had significant value creation as well that you can pursue. Athera, which I think was going after neurodegenerative diseases such as Alzheimer's. They tested their drug in a cohort of patients, and I believe after 12 days of dosing, they were able to show an improvement in a EEG-based biomarker, and we are intending to include that same EEG biomarker in our study as well. And that, I believe, at one point they had a very high, good valuation. And another company that might be worth looking at would be Innovus, which also had a very good run in its stock with its, I believe, phase 1 data that it reported on. So we are, approximately 9, less than 12 months certainly to getting into the clinic, and potentially as little as 15 months out from a major value creation event. And I believe that also at with the efficacy data in people, that's also when pharma would also take another good look at us see if there may be an opportunity to work with us. That cannot be obviously guaranteed, but that would be something that we would be actively pursuing as well. So again, moving from a preclinical to a clinical stage company quickly, but we do have another asset that I'd like to talk to you about briefly. We are also looking at redeveloping psilocybin for depression and addiction. Psilocybin, and it represents one of these novel psychedelics that have got a very strong interest recently for their prospects in treating depression and anxiety with the new mechanism of action triggering neuroplasticity. Now, one of the problems with psychedelics in general is that they suffer from some liabilities or drawbacks. In the case of ketamine, it can be abuse, and clearly in the case of some of the other molecules such as LSD, the tryptamines, such as psilocybin, there can be hallucinations. And these are often given under medical supervision because of that. So we like to ask the question, well, what if there was an agent, a drug that had the benefits, the neuroplastic effects, but did not trigger the hallucinations or suffer from an abuse potential that you could take safely at home? And I think this is the next iteration in the psychedelic space, and a good comparable might be to look at is Delix Therapeutics. They're a private company down in Boston. They just closed $70 million financing in September of this year. I think their pre-money was roughly $250 million, according to Forbes. And this is exactly what they're trying to do, is they're trying to develop non-hallucinatory, safe drugs that you can take at home so you don't need that medical supervision. And the idea of these psychoplastogens, that is, these drugs that trigger neuroplasticity, have potential beyond simply depression and addiction. There's a wide range of diseases they may be able to affect. So this is a very, very promising area for development. And I think this is where ultimately the pharma is going to come into play. It's hard to see how people will commercialize psilocybin alone in one way or another. I'm sure they're working on it, but this way is also a nice, safe way with a highly defined clinical trials and manufacturing process to get into this space. Also, again, it's the idea of patented molecules having composition of matter patents is going to be very important for pharma. So this is essentially what we've done is we've started off with psilocybin, and we've made a derivative that we believe has got many of the benefits, but a few of the issues associated with that compound. And in our initial studies, what we found is that the molecule, when dosed into animals for just 7 days, had a very fast onset of activity. These were molecules that were given orally, so that's a very important part of any product plan. They're water soluble, and importantly, no evidence of hallucinations at this point. And again, it was much better than Prozac in a head-to-head study here. So, and again, those 3 stars suggest that it's a very strong, statistically significant signal. So again, we have an interesting program that we're fleshing out, and I won't spend too much time. We're at the present building additional molecules to test as well, and our goal is within 6 months, by say Q2 of 2022, is to have a lead candidate identified for clinical development. And again, I believe that that will be another opportunity to go and approach pharma in this space to see if there's an appetite for working with us. And the little few times remaining, we do have a fairly strong scientific advisory board. Dr. Bruce Kronstein, I wanted to point out, was one of the people who discovered the adenosine A2A receptor and its effect on fibrosis. He's a world leader in that space. And Dr. David Bloom, who's a world leader on the adenosine A2A receptor and Alzheimer's disease, is also advising us. Dr. Jared Young is an expert on testing of drugs to see if they have an ability to affect cognitive and behavioral endpoints. So we're well positioned to advance our candidates and get the advice that we need for both the CNS and NASH space. Really quickly, to just go over our stock and cap table, we have just over 32 million shares outstanding. Insider ownership is in excess of 38%. Myself and the founder have about 10 million shares between us, which we purchased. Warrant coverage is modest, and there's accelerated clauses on those. Cash as of August 1st, roughly just shy of $2.5 million. Our burn rate is roughly 94 per month. And right now we're getting through our manufacturing and toxicology and are identifying that second asset in the neuroplasticity psychedelic space to further de-risk the company. Executive management, myself, and I welcome you to read the bios of our committee as necessary. And I will leave it at that and welcome any messages from the Q&A.
Thanks, Mark. We have a few minutes and a couple of questions. The first one asks, where do you want the company to be 2 to 3 years from now?
Yeah, that's a great question. So I mean, the goal of any, well, not any, but certainly in our case, a biotech would be to try and secure a partnership with a larger pharma. Sometimes that can occur at this stage. Sometimes they like to see data in the clinic. And typically it's the clinic where, like I said, you see your biggest increase in valuation. And so we're trying to get to that stage as quickly as we can with the minimal capital requirements to get to that point. So my hope would be that we would be either A, in discussions with a larger partner, or B, have increased the valuation of the company that we may be comparable to either an Athera or an Innovis in terms of pure capital markets play. So again, we want to do value creation and either appreciate the stock price or give a complete exit to our investors, either way, and likely to be on a larger exchange if we go the route of the PubCo route, and like I said, try and secure a partnership for our lead compounds with some clinical data, maybe even the preclinical stage. That's a little out of our control, but that's kind of the two areas. We'll keep growing the business as necessary, but you know, the end goal is to try and get to a partnership with a large pharma.
Thanks, Mark.
Thanks.
The second question asks, you're obviously a small biotech company, but as the president and CSO of the company, what gives you confidence that the compounds that you're working on have high chance of success?
That's a good question. Thank you. Well, again, we like to start off with a molecule we feel very, very comfortable with. So in the case of istradefylline, it's the only approved adenosine A2A antagonist out there. There are some in development. It's the only one that that gets into the brain of those that's efficiently done. So we started off with that molecule and have made conservative changes to it. So we would expect that the drug should be still fairly safe. There's always a risk that there's unexpected toxicology, but we've done our best to mitigate that. We've introduced fluorines onto our molecule, and fluorines occur in about 30 to 40% of all drugs now. So it's a fairly safe, conservative change to make in a drug. And as you can see, it improved the oral bioavailability and the half-life. So we think that the toxicology should be somewhat predictive. And based on the preclinical experiments that we've run, we feel that the drug is got a very good chance of being active in man, as is in the case of istradefylline. So those we feel are giving us some very good confidence. And finally, there's a fair bit of science saying that the target is involved in these diseases. So there's multiple groups having lots of data suggesting these molecules could have promise in those areas. So that's in a nutshell how we do it. And it's the same thing with the psilocybin. We started off with that drug. There's very good evidence. It's working right now in terms of treating depression. We've used that as the starting point. We know how it behaves and so forth. So we've made derivatives of it. Very, very similar story to the istradefylline, if not identical.
Thanks, Mark. We only have 1 more minute, so if you would like to go ahead with your closing remarks, that would be great.
Great. Again, thank you, everyone. I think hopefully I've convinced you that Marvel's a very interesting candidate, well poised to be in the clinic shortly. Done a good job trying to de-risk the drug development process and going after some very large markets with unmet medical need. Again, thank you for your attention and hope you enjoy the rest of the conference.